This method can help answer some of the important questions in the field of bioprocess manufacturing regarding aspects of cell line characterization and media and process optimization. Advantages of this technique are that it provides greater control than shake flasks and allows automation and the performance of large numbers of small-scale studies for quick data generation. Demonstrating the procedure will be Rashmika, Velugula, and Casey Kohnhorst, research fellows in my laboratory.
Begin by immersing a stock vial of 3 × 10⁷ CHO cells per milliliter of freezing medium in a 37 degrees Celsius water bath. When only a small sliver of ice remains, gently pipette the rapidly thawed cell suspension a few times, and transfer 1 milliliter of cells into a sterile 125 milliliter vented shake flask containing 29 milliliters of OptiCHO medium. Place the flask in a cell culture incubator at 37 degrees Celsius and 8% CO2 with shaking at 130 RPM, subculturing the cells after 72 hours in 100 milliliters of fresh 37 degrees Celsius medium in a 125 milliliter spinner flask for another 72-hour incubation at 37 degrees Celsius and 8% CO2 with stirring at 70 RPM. On the third day of subculture, add fresh pre-warmed medium to the spinner flask to maintain the cells at at least 90% viability for a final 24 hours of culture. The next morning, click on the remote desktop icon